Okay, good morning. So this is week 12. We are going to mention two concepts which can help you to do another level of this data mining or data science pattern recognition. These two, the first concept is manifold. It's a non-linear data point structure, and it's kind of understanding of the data structure and let's see what approach we can use to learn from these data points. Remember, the distance between data points are the most important ones. But when you have a manifold structure, the distance we have to reconsider how can we measure the distance between data points. And the tensor is another, I would like to see the more advanced data structure, or it's one level beyond the matrix, because majority of data set we play with in the data science in single cell RNA-seq on the matrices. Okay, this 2D array. You have a row, you have a columns. Well, the tensor is high dimensional matrix. Any matrix have a dimensionality beyond 3, okay? 3, 4, even higher. So how can we take advantage of this high dimensional structure of the data type and expand a lot of this decomposition method to high dimensional, and to see whether we can learn from the multiple dimensionality together, okay, instead of do one slide of the data and another slide of data, but we consider the 3D or 4D, even higher. So we will go through those concept of manifold learning for first using two examples to do this dimensionality reduction. T-SNE UMAP actually are the manifold learning tools, okay? But here we use more generalized embedding algorithm. I will mention isomap and Laplacian eigenmap. Then we will learn if you have two data sets and consider them as a two manifold, how can you make alignment to learn the relationship or inner structure among these two manifold data sets? We have applications of this manifold alignment using multi-omics single cell data. Okay, right now we can have single cell RNA-seq, we can have a single cell ATAC-seq. ATAC-seq is for the epigenetics information from the same cell sometimes, okay? We probably will have single cell protein concentration information if you apply those techniques to a same group of cells, and you assume those cells have a similar similarity structure, and how can you match them to learn their shared components using the manifold alignment concept. Then we will discuss the tensor, and then mention the decomposition method applied to tensor. Then I will give you several applications for tensor decomposition and see help you get the idea, and then later sometime you can apply to your own data set analysis. So what is manifold? Manifold is topological space which is locally Euclidean. So it gave you a framework to help you to do this supervised learning. And when I say Euclidean, that means if you look at the small scale of this the structure, you look at a local region for particular region on this object, complex object. But for the local, if you only look at the neighbor points, and that area is so-called flat. Flat means you can measure the distance, use Euclidean distance. Okay, so it is abstract mathematical space. So it's in your imagination. Well, this object in this high-dimensional space is characterized by the global structure, which is very complicated, something like this, okay? But locally, you can approximate the distance between local data points using Euclidean distance. What that means is in this neighborhood of the local resemble a Euclidean space. Well, if you move away from a local, then you cannot just use Euclidean distance to measure the data points distance between data points. Okay, when we see manifold, we assume that there are nonlinear relationship involved between data points. Otherwise, you can use simple PCA or MDS. They are same thing basically. To do this embedding or to learn their relationship among data points. But when you apply those PCA or MDS, which designed for nonlinear systems to nonlinear structure of data, you will get incomplete or distorted picture when you will do this. One example is like an S shape of this structure, okay? Well, the data points have been color-coded according to their local similarity. If you look at those blue cells, blue points, they all similar location. Well, when you move away from this local region, and then you move to, for example, in the middle of this structure, those dark red points are not so close to any of these blue points in the end of the shape. But because of its complex structure and Euclidean distance between red and dark blue points, you can measure they're not so far away from each other. But the real structure, if you expand this whole object, you should measure the distance from the blue to the red. Follow this line here. Well, this structure, when you look at the local, this local patch, okay, basically you only consider the near neighbors. They're okay. The distance between those data points, you can use Euclidean distance to measure it. Well, this blue to red, you just cannot. They look very, very similar, very close to each other in the high dimensional. Okay, but in reality, you should go follow all the structure of those points to reach from the blue to the red points. So manifold learning is try to reduce the dimension, for example, from this 3D to 2D, but keep the relationship between data points. Not only the local ones, because the local ones is easy, the distance in this embedding and in their original distance are the same, almost the same. Well, the distance between two data points, which not so local, from blue to red, the distance in this high dimensional space, if you use Euclidean distance, you can jump from here to here. Okay, there it looks like a neighbor, but you have to the correct learning, you should reconstruct or recover the real distance, okay, in the low dimensional space. So this learning is try to understand this high dimensional, so with a big D, and you reduce the dimension to small d, which is lower, but try to keep the global relationship between points. Right? There are many nonlinear dimensionality reduction or manifold learning algorithms for this purpose. So I will only use isoform, isomap, and Laplacian eigenmap to illustrate the point how this manifold learning can be conducted. There are many others as well. And this one actually is UMAP. If you read uniformly manifold approximation and projection. But when we use UMAP, we don't have, we haven't mentioned manifold yet, but the concept is all in the data scientist's mind. And the kernel PCA, okay. Any questions so far? Easy? Okay. Okay, so the idea is simple. So first you have to have this concept of manifold. So, and then to learn the structure of global structure, we start from a local. So from the local neighborhood structure. So you move small steps, and gradually you approach the longer range of distance using a series of the local steps. Okay, you don't jump, you just move slowly using local to approximate the long distance. So this method, isomap, was introduced in year 2000. This is a science paper. Okay, so this is like a spring roll structure, and they illustrate the Euclidean distance using this dashed line. Well, the real distance, you should go structure all over across the main body of this cloud until you reach another point. So the real estimation for the distance should be in this red curve. That's illustration, small example. And then you can use this network to describe this distance between neighborhood of the points. And then you unwrap this structure and look at the distance by small steps on the network. So isomap use distance matrix as the same as an MDS. Remember MDS, we have a distance matrix between different cities. Once you have that matrix, you can project the city's location on the map to let the position and the interdistance between different cities satisfy the given distance matrix. So isomap is exactly the same algorithm as MDS. So it will compute a distance matrix and then embedding those location, those cities' location in the low dimensional space in the map. The only difference is when we do MDS, we use Euclidean distance. Well, in isomap, we replace this Euclidean distance, use geodesic distance. Okay, so it's more close to the reality. when we talk about the geodesic distance, we're moving the land among the upon the landscape of those data points. So you have more realistic estimation between distance of distance between data points. Okay, then you replace Euclidean distance with this geodesic distance, and then the next step, once you have the distance matrix, the next step will be the same as MDS, multi-dimensional scaling. Okay. So in this small example, illustrate the difference between Euclidean distance and the geodesic distance. In example A, the Euclidean distance between this end and this end is given by this dashed blue line. Well, geodesic distance, you have to cross all the small hills and the small valleys, another peak, until you reach that. So you have to follow the landscape of those. So this is like an analogy example for the data points, their relationship form this complex shape. So you cannot go using their coordinates, just simply compute the Euclidean distance. So the ISO map is graph like this. So you have the local link between the data points. Okay, you don't jump from one end to another. There's no link. You have to follow those small steps. So one end to another, if you compute the Euclidean distance, use the coordinates, you get the blue line. Well, if you have to follow the small steps, you have to move to another neighbor in the network. Well, another neighbor, you have to jump to another neighbor. And this red curve is not Euclidean distance. Okay. There are multiple steps add up. When you write this example, the data form a shape of this half moon, and the A and the B, the Euclidean distance, if you project into a 1D and then from A to B into C and D, well, if you follow the geodesic distance, you go from A and all the way through the half loop and to the B. Okay, the distance is very different from Euclid. So this is a very easy, very simple idea. So this is one of example. So we have multiple high dimensional data is gave like a matrix. The question here is to compute the matrix X and its distance from X to A and the distance from X to B, and see which one is greater. Okay. When you map this into a high dimensional space, consider all the other points, and then use geodesic distance, and you measure the distance from A to X and B to X, and this gave you the conclusion that X is closer to A in reality, but by directly compute the Euclidean distance, you get a different conclusion. Similar here. So to compare this, each of the data points is like described by all the similarity matrix, or like distance matrix itself. When you consider the distance, you have to consider a lot of other matrices and form this very complex shape of the manifold. Okay, in summary, so ISOMAP is very simple algorithm. Just use MDS as a framework, as a backbone, but you use geodesic distance to replace the original Euclidean distance matrix. How can you compute the geodesic distance? And we normally will build KNN, nearest neighbor network between data points, and follow a graph, the parse, the shortest parse from one node to another, but across the graph. We will mention this algorithm next time, next lecture, next Tuesday. We are talking about the graph, talking about the network, and once you have two nodes on a graph, how can you compute the shortest distance? Shortest distance will be used as a geodesic distance in this case. Okay. That's what I mentioned. You have to compute the shortest parse between two nodes. You build a KNN, okay, the neighborhood graph, and then there are algorithms for those distance, shortest parse on the network, basically. Okay. Once you have this, the last step are the same as a traditional multidimensional scaling. On your right, this is example code example that's in Julia. So previous semester, I teach people using Julia to replace, but they are very similar to MATLAB. But this is a established package. So it's using this package called Manifold learning, and then you get the data and fit using this method. So this is not very helpful because you don't see the under the hood of the source code, but you just remember their packages to help you to do this. Okay. Okay, so we mentioned that use graph-based, okay, local steps. You have KNN network, and they are also mentioned the geodesic minimum span tree, the old method to help you to link all these data points and make them nodes in the graph, and then you move from one node to another to approximate the geodesic distance between data points. There are another way to just directly apply it onto the Euclidean distance matrix to do some kind of transformation. Well, this transformation will help you to convert given Euclidean distance matrix into a so-called affinity matrix. Okay, so this is a kind of a kernel function applied to those distance to transform those, basically change the values in the distance matrix according to this function. Okay, so yeah, I wouldn't see this very complicated. Let's see. Okay, let's see how this works. If you have a distance matrix, and for example, you have a point P is between I and J. 2 points. Okay. Okay. But we are going to use some kernel function Let it become another matrix, same shape. Will this become A infinity by J? This one-to-one. Translate. How do you translate what is function? Function Z. This function in your x-axis, so that's distance. Okay. In the y-axis, it's simply value A. Then the shape of this is this function. Oftentimes people use Gaussian curve. Okay? It's commonly used. So what is a Gaussian curve? Gaussian curve is L shape. So when you have a small distance, so this is small d, then you have a large A. When you have a large B, you have small a. Okay, so that's just a transformation, it's change the value. And you can select this shape, you can make it skinner, okay? And make it broader. Depends on your parameter. Okay. When you make it thinner, and then once you jump out certain threshold of distance, you suddenly become very small, right? Okay, in this distance. So if you use a kernel function with a parameter, which give you a skinner shape of the curve, then any distance jump away from some kind of cutoff here, then it become very small. So if you apply this kernel function and highly selective, a lot of this D become close to 0. Because you apply this, when it's cross some region, then become very small. Okay, yeah. This distance A becomes small, almost close to 0. Well, if you apply a broader this kernel, and then a lot of this D still have some values in there. So you can select, make this affinity matrix more sparse or more filled by values, larger values. So you can choose this. So when you have a lot of zeros, that means you only make link. You can make points which close to each other. Once they are another link far away, and you're going to make them different level. Okay, so somehow you create a local graph using this point. You have to be small enough when you jump too high, too far away, it's not really because we're going to move the distance. Okay. So you translate the distance matrix into affinity matrix. Affinity matrix becomes sparse only close by north funding. You automatically create a sparse network. Okay. I think the value for this is those values are not close to 0, not absolutely zero, but close. So you have some kind of soft cutoff. Do a further transformation. So, okay, so let's look at another algorithm called Laplacian eigenmap, published in 2003. The first step is also to build a neighborhood information, okay, build a graph. Same idea, so this graph generated can be considered as a discrete approximation of low dimension manifold in a high dimensional space. We look at the neighbor distance between data points. Okay, so then they will build two matrices and they apply this eigen function to find the embedding and using eigenvector to embedding this. Let's see. Okay, this is the description of the whole algorithm. There are multiple steps. So we will use small examples to illustrate, but the main thing is here. You start from a distance matrix, basically. Okay, then you apply the kernel. So hit kernel, Gaussian kernel, almost the same. You can use KNN, okay? Then once you have this transformed, this W matrix here, we can compute degree matrix, okay? We can compute Laplacian matrix. We mentioned the Laplacian matrix. Yes, I think one time they're using Laplacian matrix to, for what purpose? To do the clustering. It's called the spectrum clustering, right? So I may give you the final exam questions. So which place we use the Laplacian matrix? So you have to multiple choice questions you have to answer. There are place which is in the Laplacian eigenmap, also in the spectral clustering. Okay. Another question could be their generalized eigen decomposition. So what two places we mentioned that okay. Generalized eigen decomposition. But this is one place which we will use generalized eigen decomposition. Let's see how can we do this. For example, you have a data points in this shape, okay, in the yellow points here. We will consider them as a vertices in the graph or their data points. We will first measure, compute the distance between all the data points, okay? Then we can see, okay, those data points has to be smaller or equal to a small number in order to be connected. Okay, so this is just one way you use hard cutoff to see, okay, they are neighbors. Okay, we can use KNN. KNN, you don't consider how far, as long as they are nearest the neighbor. Or you can use kernel transform. You transform this and use some soft cutoff to enhance the separation of those. Okay. So in this example, they use the hard cutoff, the distance between any two data points. If the smaller than this given cutoff, then they are linked. So once you have this, you build a nearest neighbor network, and there are different flavors of this network. You can see, okay, there are neighbors from their distance. Okay, these 2 points are the nearest distance for this one in the corner, but not necessarily the opposite. So you can build a directed nearest neighbor. Or you can ignore those direction as long as they're neighbors depend on either nodes, you can make a link. This becomes symmetric. You can also ask, any link has to be neutral nearest neighbor. So this point has to be nearest neighbor of this point. This point also has to be nearest neighbor of that point in order to make a link. And there are different versions of those. Okay, in this example, they start to apply Gaussian kernel. And if you have two distance which close enough, then you have a high weight. If you are the distance is this two data points step away from this nearest neighbor region, which gave by this shape, then the W become small. Okay. You can change the shape of this by changing the sigma. So now we have a linked graph. And also, the graph is weighted by this W after the transformation of the. All right, so you see, okay, so in this graph is it's not a fully connected. Fully connected means each node is connected with others, but this is not. It's already been selected by this KNN step, nearest neighbor, and their distance has been computed based on their Euclidean distance and transform after this kernel function. So we have both the topology and also the weight between links. And the D is called a degree, basically all the weights of the link next to for this node multiplicate, not multiplicate, sum together is a degree for each node. And also we will compute the Laplacian matrix using the difference between degree and its weight, link weight matrix. All right, so these two matrices, they all square the matrix. So D depends on how many nodes, how many data points in your data set. So D will be n by n. L also is n by n. So the two squared matrices. Then we are going to apply so-called generalized eigenvalue problem. Okay, so L and D are two squared matrix. We will find F. Okay, F is eigenvector set. Okay, lambda is the eigenvalues. Okay, so this small box, this insert, are the screenshot for MATLAB function, which function which can take two matrices A and B. So A and B in our example is L and D. So this function will help you to find V and D. V is eigenvector and D is eigenvalues. They will satisfy the relationship when you use A multiplied by V, the result is the same, equal to, okay, B times V times D. So this formula here, okay, match the shape, okay, match the corresponding values of this equation. So this is called generalized eigenvalue problems. So the information we use, including Laplacian matrix and the degree matrix. Degree matrix tells you the connectivity between the values for each of the nodes in the graph. Well, L matrix contains information between nodes. Okay, once you found the eigenvectors, which in the F, you can start to use F, which corresponding to the second largest eigenvalues, and use the following F as a, to use the following eigenvectors as your new corresponding position for the data. So that's called a projection, something like similar as PCA. Okay. So you replace the original position of the data using eigenvectors position. To complete this dimensionality transform, okay, from the high original dimension, you learn from the relationship between them, and then you compute using generalized eigenvalue problem and found the vectors. Those vectors combined is a projection of your original data points onto the lower dimension. Depends on how many eigenvectors you use. You can use the two eigenvectors, then you project your data point to two dimensions. You can use three. Okay, so that's why you come from the 2 to the D plus 1. Okay. Yeah. So this is a toy example using only 4 data points to illustrate the major steps. And the example is provided by this YouTube. So I took some screenshot. Okay, once you have the location, you can start to compute the distance, and then so you compute the distance for between all the data points from 1. Let's see. Okay, everything is fully connected, and then use the nearest neighbor, and you filter out, get rid of those some of this, and that's your KNN network, nearest neighbor network. So the weight between all the data points are given by just 1. Okay, so this use a very simplified version of the weight matrix. Every link have a weight equal to 1. And then because 1 and 2 are linked, so the position 1, 2, their value, and also 1, 3 are linked. So position 1, 3 in this matrix are linked with 1. Other places are all zeros because there are no link between 3 and 4. 3 and 4 is here, so 0. Okay, so this is a weighted adjacency matrix. Then you compute degree matrix, like sum all the columns here and put in the diagonal position. Node 1 have a 3 linker, so the degree is 3. All other 3 nodes have a degree 1 because they are only linked. There's only one link in the graph. So you have these two matrices, then the difference is Laplacian matrix, and then you plug these two matrices into this function to get the generalized eigen decomposition solved. And then you just use those F, which is the eigenvectors as your new embedding position. Okay. This is source code in Julia, very similar to MATLAB. That's MATLAB solution, and the value is slightly different from what they provide. I don't know what the package they use, since we have a slightly different solution. For the embedding. Maybe, so this is from 1. Yeah, lambda 0. So they use this index start from 0. So lambda 1 indicates the second largest eigenvalue. So that's the second largest eigenvector. Well, in my case, what I got slightly different values, but the idea is you reduce the dimension from 3D, okay, 2D, this graph into 1D. Relative position of those nodes are project onto this 1D space. Okay, so this is a very small example, and it's nothing as a good illustration for a non-linear relationship among data points, because it's 2D, nothing. But the whole idea or whole pipeline is the same. You just replace what you have in a more complicated data set example, but the steps are the same. In reality, there are algorithm linked between Laplacian eigenmap with PCA. So the PCA, so you can consider Laplacian eigenmap as a more generalized version of the PCA. PCA only consider two matrices. You only consider the covariance matrix. There's only one, okay, in there, but a similar idea. Okay, so this graph compares the embedding algorithm and the way you apply linear PCA onto this Swiss roll dataset. PCA will not be able to separate the data from the very clearly, and because it's supposed to separate blue points with red points because they are in a different location in this Swiss roll structure. While other algorithms are designed for this nonlinear relationship, they can recover the topological global, so-called global topological difference between those data points. For example, Laplacian eigenmap give you one end are those red points, another end is blue points. Well, PCA will project everything onto each other. Okay. So Yeah. These are the other examples, some toy example synthesized a shape like this and another shape with S, and also their local structure within the S shape. The more complicated is one and that one. And those are the nonlinear manifold learning algorithm that give solutions, slightly different, but capture the majority of the global structure. This is important. You consider single cell data, okay? So you have a cell type, this and that. When cells, there are different cell type, but when they all from, for example, in the G2 phase or in the S phase, they probably look from a certain angle, they're similar. So if you don't consider this nonlinear relationship, you probably will cluster a group, two group cell types into one cluster. So those problems have been solved using T-SNE UMAP. Remember, UMAP is manifold learning algorithm. But this lecture just gave you the concept that we have to consider the relationship between data points in the more complex global structure. Okay, when people use in their paper to describe their algorithm, say, okay, the fate, we are going to discuss the fate algorithm can generate embedding of the data points in low dimensional space, but keep the global structure in the higher dimensional structure better than other algorithms. So you know what they are talking about. Okay. Yes, let's take a 5 minutes break. We will come back to look at one of the application in single cell data analysis. We mentioned this, it's called a fate embedding. Okay. Okay. Okay, so this is kind of review and the comment paper, and so this group is in the Yale University, SMITA Krishnama Sawama and her lab, her postdoc graduate students are among the pioneers who applying manifold learning and to create a lot of the manifold learning based method and for applications in single cell data sequence analysis. They so you we mentioned once this embedding algorithm called FATE, and so we have t-SNE, UMAP, and FATE are the top three options. Normally, we will do in the single cell embedding. So that's the first step. Normally, you after the QC, you want to reduce the dimension and put the cells in the 3D space and look at their relationship. So so they consider those cells follow. So the cells, okay, in the single cell data set, they follow this would like to see if you consider each cell is a data point, then cells are linked. They are under different of this development trajectory because of this cannibalization effects. They don't jump from one state to another. They always have some kind of neighborhood linked together. But globally, when they fully development, they developed into different stages. And among those developed states, they are not communicate or not in the same states. You have to move back and follow their development path and come back to this trajectory. So this automatically form this complex structure then we simulate a model use manifold. So their idea is to look at the local distance relationship between all the data points and then create this local nearest neighbor structure. So what they start with, for example, if you cells from this shape in the high dimension, and they start to compute the distance between all the cells. So if you have 2,000 cells, your distance matrix will be 2,000 by 2,000. Then to keep those distance which close to each other enough, they apply this kernel transformation. Okay, if distance too far, and then your affinity will be lower, right? So if you have a low affinity, you will be removed those link. If your distance is close enough, then you have a high affinity, then those links will be kept. So after you finish this, use this kernel to do the filter, and then you end up with this nearest neighbor graph. So you see this is fully connected, everything connected with the other. But after you apply kernel function, those links between distance related points will be removed. So you end up with this KNN network. Okay, only keep the nearest neighbor basically. And this neighbor, the matrix described this network is more sparse than this. So this matrix, all the 2,000 by 2,000 cells, the distance, they all linked, and all the position in the matrix has values. Well, after you filter them out, and only the cells which are the neighbors contains their affinity values. the rest of them is set to 0. So that's the idea about this fully connected network with a lot of zeros. Okay. So once you have this network, you can start to move from one cell to another. For example, if you move from this cell to this, you have to go through all the way across the network until you reach out by the small steps. So through that process, you actually learn the manifold, underlying manifold structure, okay, by compute the distance, the steps from one cell to another, from each cell to another cells. What they found, they can apply this method to re-establish pseudo trajectory, pseudo time analysis, better clustering, because you separate the cells by their similarity and also their relationship with others globally more clear. Then to do the denoise, okay, visualization. So basically, if to denoise, you can use one gene, one cell's expression to correct other cells' expression by looking at their nearest neighbors. So once you have this denoised data, you probably can recover the relationship between genes, okay, because all the cells, the value has been denoised, and those points on the cells and the relationship between genes may be clear, and so it will help you to construct gene regulatory network between your genes. So manifold learning concept also has been used in most of these pseudo time methods. We mentioned monochrome 2 and the slicer wishbone diffusion pseudo time. So they all have some of this KNN in the middle. So KNN, the purpose to build a KNN is to use the local steps to approximate the global structure of the data. This is from the same Smita's lab and the same lab from the who produce the generate the fate algorithm. So this is called magic, is imputation method. Try to help you to denoise the matrix, okay? That's before the magic imputation and after. Okay, in the middle, the process is the same, almost identical to the fate. So identical to the fate means every step here in the column B is the same until the last step. The last step, instead of doing the embedding, they try to reconstruct the expression matrix. So basically, you have a input expression matrix, you will get output expression matrix the same size, but a lot of values have been filled in. What they found is in some situation, because the remove of those zeros in the sparse input matrix, you get a better correlation between genes. Okay, it was like this. There's no way you can see, okay, gene A expression level high, gene B expression levels also high among different cells. Because there are so many zeros, so a lot of values actually collapse either in the x-axis or in the y-axis. Well, after using magic imputation, because all those zeros have been replaced with some small values according to the neighborhood relationship among cells. So you actually recover a better correlation if these two genes are positive or negatively correlated. So you see this, because all the cells has values, okay? You start to see the correlation. So after you're embedding this, they can find the cells in the tips of those structure to learn what's special about those cells, to learn this trajectory. Okay, those this corner, the characteristic feature of those corner. So each corner may indicate or imply some of the direction of the whole population cell development trajectory, a trend at which direction they want to go. Though that's also help you to do the clustering, okay, better understanding of the different clusters, because all the values of the field, zeros, so all the negative impact of the zeros, the dropouts, didn't remove. And they also have some examples if you read their original paper, because you have values, you get the power to understand the relationship between transformed factors and their targets, because the correlation between them can be more readily established compared to the vectors full of zeros. Okay. This is just indicate that we can do the KNN network for the spots of those spatial. It's not very important. Okay, so manifold learning is also the manifold-based machine learning algorithm. It takes two more than one data sets, okay, between sides of data to produce the projection. Okay, so you have two data sets, and then you believe they are shared manifold, the common shared manifold structure. They project both data sets onto the shared common manifold to make them align. Okay, although these two data sets may compose of this from the different measurements of the same project, maybe. So something like this, you have a teapot and rotate horizontally, and then you have this small object as well, and they share some of the structure of their movement. So you can align this. And then this characterized by the same underlying mode of variability. Okay, so you trade this movement, the shape of the data collected from different angles, and project this object and this object into the shared manifold and make them aligned. So you can put this and the corresponding frame of this data. It will make a link between them. Okay. So this take advantage of assumption that these parent data sets produced by the similar generating process. So you have different processes, and then for the same samples or same group of samples, the generated information is shared similar underlying manifold representation. So we have to learn this manifold representation, which is in the middle of these two data sets. Okay, so these two data sets share this structure with order of different data local connected. So we try to get the position, for example, for this so-called shared manifold, or the data points in the position, and their correspondence are recovered, and knowledge from one domain can be transferred to another. Okay, So if you have this object, and once I give you a point from X domain knowledge, you can map it to here, and you know, okay, where they come from the Y domain. Okay. So this small example of the 3D object. This actually is protein structure, basically. But these two protein structure files are in the very different scales. Okay. The right one is much larger than the blue one, but their coordinates largely the same. Okay, so this if you give this position of all this data, one from red, one from blue, to the algorithm will do the manifold alignment for you and to find their shared position after you map them according to the shared position they matched to each other. So people borrow this idea to try to map align this manifold, which data from generated from a different technique, for example, for the same biological objects. So this is one of the algorithms. So this technique is useful, as we said, the multi-omics technologies can produce different kinds of the readouts from the cell. Okay, now we are talking about same group of cells, same sample, but not exactly the same cell can be measured into two techniques. Although there are techniques that you can measure in the same sample, different technique, same cell, exactly the same cell, just like what we discussed last week. It's about surface receptor type. There, sometimes you can get, but most of this you dissociate the cells and for the same population, same sample, and gave to different technology. In this illustration, on your left is RNA sequencing for the mRNA, so this transcriptome data generated. On your right side is epigenetics information. The most commonly used epigenetic measurements we can have is called ataxic. Who have used this or no? Everybody knows ataxic. You don't know ataxic. You have no idea what it is. Okay.
Heard about that, but what's that?
Okay. So you know, for the higher eukaryote organism, you have heterochromatin structure. Okay, so you have this long linear DNA on the chromosome, but some region is highly packed. Okay, this is called closed. And some place the DNA is more exposed to the environment. And this process is kind of dynamic when you need the region, for example, to let this transcription factor protein binds on it and to start the transcription machinery. This particular region can become open chromatin. So across the genome, on the chromosome, some place is highly packed, not ready to be regulated, where some place is open. So when you people want to know which under the cell under which condition, which part of the genome is open. Open oftentimes means their regulatory elements start to function, and if you know, okay, what this regulatory element regulate which gene, so you know, okay, which gene is ready to be highly expressed. So people want to know this dynamic change of the open chromatin structure. So one of the techniques they found that they use this called transposis. So TN5 transposis is this enzyme which extracted from the bacteria or some species. What they found is this transposis has very unique features. Once you have open chromatin structure, it will cut your DNA into fragments. Okay. In the same time, they will label the cut points of the DNA with their some of these unique figures, small barcode. So people just borrow this system from a bacteria, and then see, we can use this system to cut the cells chromatin. Okay, when you collect all the DNAs, let it cut, and then we sequence for these tags. If you found the region which have a lot of the tags, is highly the place is completely open. Well, the closed part will never get cut, so you don't have a label of that. At the end, you will count how many labels has been put in the different regions of your genome. You get this picture, some kind of ataxia peaks. Because if you have from constant one, and this position one, and then you count how many those transpose tags in there. Picture. So the higher fragments will widely open, and then you believe cell at this stage in your sample chromosome 1 is condition is open, and then you look at their downstream gene or cellulose base later. If this is a promoter of the cancer region, so indicate this downstream gene, and there's regulation likely to be as well of the more active. Okay, so we want to get a taxing fix as well as this downstream gene expression. So a taxing plus real gene expression give you the whole picture of the regulation activity of the downstream gene and outcome of the field. So if you found things where this will become high and this will become high and express, or sometimes this and express and this become weak, or other way around, maybe this peak already showed up and this gene and this control has not been expressed. So you know this dynamic started get classified in more dynamic regulation between of this process. Okay. So now we have a technique which can do this for the single cell. Okay, so isolated cell, you have all the DNA. Okay, so you apply this transposes onto the DNA to get the peaks for an individual cell. Okay, so instead of RNA sequencing, you use taxi. In the single cell data, you get cells. This is right. So in a taxi, we get cells. Cell, this is cell 1. You get the two information. Some paired unique. Okay. Also, this solid of cells. And each cell has a peak. In this case, you know the name of genes, you know where they are located. In this case, you have all the peaks, you know where they are located on the console. Which cell express which peak. Not express which cell contains what peaks in what position. So ataxic is applied for the extracted nuclear. So it's in this case, they are not cells, they are nucleus. Yeah. So it's so if you have single cell data and also single ataxic data, they collected from the same population of cells, which that's half. And then this satisfied a condition which we can apply manifold alignment. Remember, we see, okay, these two different data sets are generated from the same process of the same sample, although they are not the same cells, not the same nuclear. You can apply manifold alignment to align you single cell gene expression profile for the cell against the cell which have a corresponding peaks. Okay, so if you have a cell like this here, cell 1, and in this nuclear 2, and there so in cell 1, you have this gene expressed, this gene expressed, this gene expressed. This profile will be aligned with a peak profile. And then another cell will align with another cell. So if your global structure are the same, you can form, you have southern cells form a global structure. Southern cells have no global structure. It's aligned base. You can find the corresponding cell and the peak profile to help you understand this. Okay. You can use single cell ataxic alone. So you can use this as a, it's not a gene expression, but it's just a peak profile. You can reconstruct all the cells cluster. Just treat them as the same matrix. You don't know, you just different information. Okay. Normally, you will construct the same of the cluster as you use single cell RNA-seq applied to the same population. Okay. So this illustrates the aggregation of single cell RNA-seq. In here, you use 882 cells and to reconstruct the peaks. And the peak very similar to you use bulk ataxic or use several thousand of the cells. So that's indicate the aggregated hundreds of the cells, nuclears, recover basically the whole population of the peak dynamic. So they're highly dynamic, but as a group, there are certain signal which is always there, the same shape. Okay, do you know understand about this? So that's the one of the application in the manifold alignment we can do for the single cell ataxic and the single cell RNA-seq. But nowadays we don't have to do it. The reason is we can get this ataxic and RNA-seq for the same cell, so you don't have to align them at all. You know the ground truth. Okay, it wasn't possible like 2 years ago. This is very new. Okay, very new. So before that, people try to establish this manifold alignment to match the possible cell states and measure it between two techniques. But now you don't have to. You have a direct link, okay, the cell expression level is like this, and for all the genes and what their ataxic peak look like. It's a very powerful way to. We have some example data, but we don't know how to use this information yet. Okay, so this is like a data scientists still try to work on how to use this matched single cell ataxic, single RNA-seq to learn the regulation between genes.
For the ABC, this kind of I said maybe it's better to have maybe an apple that's called the chromosome.
To understand what high-C?
Just the structure of the chromosome.
Yes, we are talking about the different, slightly different. You see the 2D or 2D structure between chromosome. So the fold and sometimes the interaction. Yeah, so I don't think you are talking about the high-C.
Yeah,
High-C is another, yeah.
I think slightly different.
High-C is also some of the card using with transpose enzyme, maybe, but you have to have a two different regions of DNA. They fold into this contact structure, and so you try to use high-C to understand. But I don't think we have a high-C single cell data.
But I think this I think is belongs to epigenetics.
Yeah, it's epigenetics.
Maybe it's better to combine with something else, methylation.
Yeah, we don't have a single cell methylation yet in the commercial package, but some labs claim they can do single cell methylation pattern. Okay, single cell methylation pattern will be, the data will look like very similar to those peaks. So, but that different means. But ataxic to its peak tells you the open chromatin position, with methylation peaks tells you, okay, this region is highly methylated. Okay, you may get a profile like this, single cell level, yes. But how can you use that information to tell you the biological question? That's another story, okay? Yeah, it's very difficult problem, I can tell. So now still, all the labs are busy in generating data, but very few of them can really use the information we collected, okay? Okay, in the future, we also have a protein, we have a single cell level. Okay, you see methylation, you can have a genome. You probably can detect this CNVs, copy number variation among each individual cells, especially in the cancer study. So you have the full spectrum of those information from a DNA, from methylation, from open chromatin, from a lot of other chip-seq. You have, of course, gene expression and the protein expression level for individual cells. But how can you do the integration? A lot of people claim they can do, but we need to have a better ways to understand this. This is a topic which I think the single cell world will be busy in the next few years. So this is from an early stage of application, and the kernel manifold alignment for the domain adaptation. Just some papers I threw out. And this is the application for manifold alignment to to ataxic, okay, chromatin structure and the gene expression, and also have some time series information in there. So at that time, this is an early paper, not it's not a single cell level. Okay. Their approach proposed in the Manifold's lab in Stanford is called a coupled NMF, near non-negative factorization. So you consider two matrices at the same time. So this is a single cell expression. So this single cell ataxic. They try to do this NMF by shared parameters between two of these matrices approximation. At the end, you want to find a similar structure among those cells. Okay. They are not the same cell. Okay. But as I said, this method, we don't have to use it because we can get the information from exactly the same cell. So you don't have to find cell groups which align. There are other possibilities, and I share some of those paper links. And the Surid package at that time have an extension of those analysis we can apply to single cell chromatin dataset. That was a Surid 3. And I believe today they announced the Surid version 5. Okay. So at that time, they start to claim their functions can help you to do this integration of the cell analysis from multi-omics. Okay, so we finish this part for manifold and alignment. Now, any quick questions? Okay. So we jump to tensor, and the tensor you can consider, for example, rank 3 tensor is a 3D array. So we move from a matrices, vectors, to a high dimensional data structure. And this is 1D, 2D, 3D, and even higher dimension. Okay. 3D is 4D. Why we want to use that information? Because in our collected data, there are a lot of examples. We try to collect high dimensional data, although we can flatten them and lower dimension by, for example, convolution or other range of the vectorized data. But if we have a good way, can take this high dimensional dimensions into account, that will be really good. For example, the image contains 3 colors coding, right? So each picture actually have X, Y, and it's a 3D data set. And this time series, and each picture is a 3D, then you have a time series. So this is like a 4th order of tensor. Okay. So the tensor as a variable actually has been widely used in the physics. For example, all those parameters in the those equations are actually tensors, contain the position, and also the time space. Okay. So so-called tensor-oriented code, so we can treat the variable as the same as the matrix if you language, your computer language will take care of those. For example, in MATLAB, we can define this 3D data very easy. And a function like random, you remember R. Function, if you say, okay, I want to use 3, 4. This will create a matrix in 3 and 4. Okay. Random values. And if you do see and 3, 4, 2. We will try. Remember, it's suitable. You will create. You will create another. This way. One side. So become a tensor. I have a height. So this wire. So this function is called a tensor origin. You can generate tensor very easily. But depends on your language. If you are using C, you may have to write your own library or function that can take care of this tensor structure. So you can call it number array or 2D array. Then it's called scalar, vector, matrix, just synonyms. And then you call it ND array or ND tensor. So, different word, sometimes it's interchangeable keywords. So the tensor decomposition is a very useful mathematical tool for data analysis. And let's take the same idea as you do this matrix decomposition. For example, you can decompose this big X matrix into the outer product of two vectors. Okay, so if you have a vector n by 1 or m by 1, 1 by m, and then outer product of these two will create an n by m matrix. So you can expand this into a 3-way. So you have a n by so use a, b, c, 3 vectors. Okay, so A and B create a matrix, and when you multiply by another one in that dimension, you get the whole qubit of this X. So you can apply this to even higher. So you have 1, 2, 3, 4 dimension, even higher. So same idea. So in SVD analysis, we can decompose this A matrix into U sigma V. Okay, this U and V are the smaller matrix, or even just one vector is okay. You can construct this. Same idea if you can decompose a data structure like this into this small matrix, this small matrix, this small matrix plus a small I see this is like still a small tensor, but this empty tensor only contains value in their diagonal position. So it's just a scaling factor. So you can basically the products of these three, they become the original data. So you can learn from the original high-dimensional data to learn this structure, the values of those small matrix. The values of the small matrix will tell you the components which can be used to reconstruct the original data. And in the matrix decomposition, we know we can use top matrix vectors. For example, this 1, 2, 3 is use top R vectors to reconstruct or approximate the original data. For the purpose of the denoise, for example, we only use the informative components to reconstruct and to get a clean data reconstructed. Similar idea here is to learn the original data to learn their major components and use major components to reconstruct the matrix. And this matrix is approximation of the original matrix. But if you select the correct major components, you make a denoised version of the original. So the algorithm you will have is something like this. You have IJK, the 3D stuff. And you want to make this the difference between this tensor and this tensor and their sum squared as small as possible. Okay, if you have two identical tensor, the difference between them and the sum of squared error will be 0. Okay, so your goal is to select those vectors, to compute those vectors. Once they construct the approximation, this approximation is as close as your original data. Exactly the same idea of our matrix decomposition. Okay. There are two major algorithms. This canonical decomposition, also called CP, and they independently reinvented twice in the 1970s. So CP sometimes called different names. CP, okay, CP method. It is an optimization problem. As I explained it, you try to minimize the difference between these two tensors, original data and reconstructed. Original is X and reconstructed the M. M is reconstructed by 3 A, B, C, okay, 3 set of these components, vectors. So this is the equation for the minimization. You try to minimize this difference. So the method people normally will use is called alternate least squares. So you consider just one step for one of the dimension, and then you step 2 will consider the B, okay, second dimension. And step C, you optimize the values for C, okay, and then you turn back to ABC again. For each time, you just try to minimize, try to fix values for one dimension, and at the end you stopped. There are some problems, some tricks that you can make this process easier. One of the technique called randomized matrix least squared. Okay. As you can imagine, this will take much longer than to do the decomposition of the matrices. And application is a low rank. Okay, so. Another method is called the Turk lower rankedness. There's another method to do the tensor decomposition. You see the structure looks very different from the CP method. They will construct several of these what they call this should call vector sites, something like that. Okay, so have some applications. This is to simulate to capture the dynamic change of the network. So the data was collected from a real life is a school, and then at a different time points, the binary interaction between students. Okay, and then once you have this time series network stuff, you can decompose it into red, green, blue, black components, and each of these components, each rank gave you some information about the interaction of the dynamic interaction network. I can't see exactly what's the values in there, but you can visit YouTube and to find some information. So I'm going to give you application number 2, which is from this mouse study, and then also the time series and collect the information about this neuron activity on the brain of the mouse when they expose this cross maze. And then you learn from this high dimensional data because you have time series, you have a starting position of the mouse, you have multiple samples, collection of the neuron information from multiple times. So to do this decomposition, I think I will play the video, let the speakers to explain this to you. It's an interesting story. I will find this link.
Now I'm going to motivate for the number of victims.
Okay, so that's
Turned out to be very easy to handle in some form, like I mentioned. The interesting piece is it also introduces zeros into part of the gradient computation wherever there's missing data. I mean, we sort of get this sparsity in the gradient computation. This turns out to be something we can exploit if we have very large scale data and we want to use, say, a stochastic optimization method to solve. we can actually get a sparsity in the gradient computation, which makes it much faster to compute. We have this mouse with ears. So 2 years of graduate student postdoc work, they make this lovely set of numbers out of this measured image.
Sure.
And that's what we start with. So for 300 neurons, they have sort of an activation level for every neuron. And they do this over time as the mouse is moving throughout the maze. And so we have a neuron by time matrix where we have the activation level of each neuron at time. That is one trial of the mouse going through the maze. Then they do that over and over and over again, 600 times, because they want to understand learning. So then it becomes this question of how do you analyze this type of data? So let me tell you a bit more about the experiments that they did here. So we have this mouse will start in either the west or the east of this maze. There's a wall in the middle, so when it gets them to the middle, it has to make a turn. And if it makes the correct turn, it gets a treat. And so at first, it should always turn south, whether or not it starts in the east or the west. Otherwise, so he goes along and he does that for a bunch of trials, and they change the rules. And isn't that just how life goes? The rules are just changing in the middle of your life, and then you have to switch your behavior. So then the mouse can turn right. So nothing changes if the mouse starts in the west. If it starts in the east, it needs to turn north to get its treat. Then they change the rules again, and the mouse can figure out a new set of rules to get its treat. So that's the setup. So we get something out of tensor decomposition that looks like this chart. So now you're thinking, I should have sat closer to the front. You can feel free to move. So what is this thing? So we're making this tensor decomposition, which looks like this. And so let's just look at the second component. This first plot over on the left is the activation of each neuron in a sense that corresponds to the vector in mode 1. Then we see a profile in time, which corresponds to the sort of activity in time. And notice that this curve is smooth. We did nothing to make this smooth. This is just inherent in the data. And then last is this funny business with the different trials, which are coded with different markers to say what's going on. So it's orange if the mouse started in the east, green if the mouse started in the west. Those are sort of equally spread out. Then it's a circle if it ended in the south, which is where it ended most of the time. And then it's a square if it ended in the north. And then it's filled in if it got its treat, and it's empty if it didn't get its treat. So, do we see anything here? Well, let's zoom in on this third factor here. And you can see in time, it's happening sort of towards the end. And then there's a very clear separation. I can draw a line between the filled-in circle, the filled-in markers and the not filled-in markers. So my guess is that these are the neurons that have to do with whether or not that mouse got its treat. Oh, I call those happy neurons. If we look down here at this second to last one, this is sort of towards the beginning of time, and in fact, you see a very clear separation between the green and the red, and that, the orange and the green, and that corresponds to the starting position. So these are the neurons that somehow understand where the mouse has started. If we look at the last one, this is towards the end, and it's a little bit hard to see the shapes, but the tops are the squares and the bottoms are the circles. And whether or not they're filled in, they're still sort of separated here. Squares are always on top, that's where the mouse actually ended. Circles are on the bottom. And so this is the sort of set of neurons that understand where the mouse ended. So this is very interesting. The tensor decomposition knew nothing of the conditions of the trials at all. And yet somehow it saw this underlying behavior in this low rank structure. So this is sort of the utility of these methods. And the neuron people are extremely excited, and the paper about it on bioarchive and the rest of the slides. So how do we actually compute?
Okay, so this is the one of application. They coded this different ways to look at when the neuron become active and associated with what outcome of this consequence. Okay, so last slide, I don't know if you still remember this. So we use tensor decomposition and the manifold alignment built into our pipeline to compare two samples Okay, I will try to build something which help people to go beyond the differential expression analysis. As I told you before, I'm against differential expression analysis for single cell data. So if we don't do DE analysis, what we can do? So this is one of the application. We construct gene regulatory network for 2 samples individually. Use tensor decomposition to decompose and reconstruct the network to denoise. Okay, we get 2 beautiful versions of the denoise network. We use manifold alignment to align those 2 networks collective from 2 different data sets. And those can be aligned, those genes are the gene less important, which those genes cannot be aligned very well or distance are far from each other in their corresponding projection. Then those genes are the most important gene to tell the difference between these 2 samples. And we found also those genes are the differentially invariant genes into different networks. And those genes really important functionally. Even though some of the genes are not differentially expressed at all, they are just change their partners in their corresponding networks in either sample 1 or sample 2. So we detect those network rewiring events and found those genes really tell you the importance of this difference between these 2 samples. So we will play with those examples in computer science, okay, computer lab Thursday. Okay, thank you. That's all for today.